Welcome to the lab, HSJ Hylton. In today's session, we're focusing on HSB and in particular, the topic of digestion. Now, very important to recall, what we need to remember is the actual substance that is digested, the enzyme acting on that particular substance, and what it is broken down into. Now, we have two forms of digestion, mechanical and chemical. Mechanical digestion involves the breaking down of large food particles into smaller pieces by way of the teeth. Chemical digestion involves the breaking down of the food particles themselves from complex substances into simpler molecules. Digestion begins in the mouth. In the mouth, we have the digestion of carbohydrates. The enzyme acting on carbohydrates is called amylase, otherwise called ptyalin. These break down carbohydrates mainly to glucose. In the stomach, we have the digestion of protein. Proteins are acted upon by the enzyme pepsin. Pepsin works at an optimum pH of 2.1. Present in the stomach is hydrochloric acid, which allows for the optimum pH of 2.1 to be maintained in the stomach. Proteins are broken down into what are called amino acids. Now, once the food passes the stomach, we enter the duodenum leading into the ileum. Here we have attached to the duodenum is the pancreas. The pancreas will secrete the pancreatic juices that will continue the digestive process. The pancreatic juices involves the lipases, proteases, and the carbohydrases. The carbohydrases will work on carbohydrates, the proteases will work on proteins, and the lipases will work on lipids. This is where we have the breakdown of fats into fatty acids and glycerol by way of the lipases that has been released from the pancreas into the duodenum. Also present there, attached to the duodenum, is the gallbladder. The gallbladder will secrete bile. This will serve to emulsify fat, which will allow for the effective digestion by the lipases. Now, once these digestive food particles leave the duodenum and the ileum, it enters the small intestine, where the absorption of these molecules will take place across the villi into the capillaries surrounding the villi. Here, the digested food particles enter the circulatory system from the digestive system and so can be carried to each cell where they will serve their function. Now, very important to note is that once these molecules have been absorbed in the intestine, their first destination is the liver. The liver then regulates the amount of each that will be distributed throughout the body. Digestion essentially ends in the small intestine, and here also ends